Okay, we are recording. And all right, welcome to master's level bioinformatics. Um, I know at least for three of you that you have not had a bioinformatics course before. Ms. Thompson, is this your first bioinformatics class ever?
Yes.
Okay, so 100% of the class has never had a bioinformatics class before, which tells me kind of how to teach this course. So, bioinformatics is weird in that at this point, I am assuming that all of you know all of the relevant biology that you need to know. That's kind of a weird thing to say because you have more biology classes. But I'm assuming that you just kind of basic biology at this point. Um, that means really the bulk of what we're going to learn in here is the informatics part of it, the algorithm stuff, the computer stuff. Um, and so that's going to make this class different probably than any other class that you've had before. Um, also, usually when these classes are taught, they are taught with programming as a component to it. Um, I'm gonna ask this, although it's always the same answer every year. Do any of you know any computer programming languages?
No.
I mean, like, beside Microsoft Excel and that whole office, besides that,
Oh, well, Excel almost kind of comes close to counting.
Um,
I'm
is
talking,
that something
yes.
like HTML or something like that? Like C++?
Actually, that's C++. Yeah, HTML is kind of also close. It's not a programming language. It's what's called a markup language. That's kind of close, but HTML is like what everyone learned
I
back in the day with MySpace.
They made me use it in high school to like create a web page and
Yeah,
made me cry every day.
yeah. That sounds about right. HTML really just tells the computer how to display information. It tells it how to show a website, which is like useful thing to know. But no, C++, or even just C, that would be a programming language. Usually when we do this class, there's at least an intro to programming, and that's sort of how I'm planning it. Currently in 2023, Python is the most used programming language for bioinformatics. And so that is what I want to introduce all of you to throughout this course. And that is why, if you read that email that I sent out, the introductory email, it said it's not required that you bring like a laptop or a tablet or something to class, but it is encouraged. Because if you have something that you can bring, you can kind of follow along when we do the programming. That's the type of thing that I think works better in person, and so I'm saving like conceptual stuff for the online lectures. I'm saving programming introduction to like when we're in person. Um, so for this course, this is the first time it's being taught at the master's level at Kaiser. And so I kind of don't know how it's going to go. What I want is for there to be a programming assignment that all of you complete. Um, Gonna kind of just play it by ear. And when you look at the syllabus, you'll see that there are no real assignments listed in what is due. That's because I just kind of want to see how things go first before I start assigning things. Bear with me. It's the first time doing it at the master's level, so I will grade accordingly to this being a little bit of a trial and error. All right, so we want to get some Python introduction. I also should probably go ahead and show you the syllabus. Let's do the standard first day stuff. So let's say share application screen. and share the entire screen just because it's easy. And there we go. All right. So no, you don't need to see everyone's grades. Oh, by the way, you probably don't care, but I finally updated my profile picture. I don't know if this makes me look my age or if this is just creepy or what. My updated profile picture. My other one was like way too old. I looked like a baby anyway.
Okay, that's not you, Dr. Cooks. You're catfishing us, just to let you know.
No, that is seriously me. I took that like 2 days ago.
Yeah,
I've
catfish.
got
I'll see you on Saturday.
Yeah, I've got a little bit of scruff now. I got a little bit of facial hair, but so that was my creepy updated profile picture. Let's go over here. So for the syllabus, setup, if the same way the other classes at the master's level with me have been, but I know that for two of you, you haven't had, this is the first class with me. So it's set up to where we have 3 tests and 1 final exam. The tests we're doing in person. And so since we are meeting every other Saturday, we're meeting like the odd numbered Saturdays. The first test won't be till week 3. So we'll do test 1 week 3. 2 weeks later, we'll do test 2. 2 weeks later, we'll do test 3. Dr. Canfield and I have been swapping it out this way to where I do the odd numbered weeks, she does the even numbered weeks. The weird thing about that is that means the final week, always for a master's class with me, we don't meet in person. So I have been doing take-home final exams. So like we just did biostatistics. That was an actual like project that I had the students do and then turn in at the end of the week. For this class, it will be, I don't know, some type of take-home final exam that you'll have like the whole week to do. The tests are 15% of your grade. The final exam is a quarter of your grade. And then there's just kind of the miscellaneous. 15% is in the syllabus is formative assessment, but that is essentially like quizzes and then any other in-class assignment that I just kind of give. We always need something that sort of counts as the presentation. Essentially, there will be at least one day that we meet in person that I have all of you get up and talk about something. It is not necessarily like a formal PowerPoint presentation. Biostatistics, it was just you have analyzed some data, get up and tell me what you did specifically and why to analyze the data. But there'll be at least one day where you're kind of graded on showing to the class something. For this class, it may be, assuming the Python programming goes well, it may be just show me the program you wrote and explain what the different parts of it do, that type of thing. All right, so that's sort of where your grades are going to come from. I have the tentative schedule built out right here. Oh, by the way, too, for the master's classes, we don't do required textbooks. We just do suggested textbooks. You are in no way required to buy this. It is a good read, though. It is suggested that you get it, but you don't have to. All right, this is showing then every Tuesday, 9 to 11:30 a.m. We're doing the online. Every odd numbered week, then we're doing the Saturday class, which goes faster than I expected for a 6-hour class. All right, so we've got all of that. Other than that, if you look through the syllabus, it's the standard syllabus stuff in terms of there is a dress code when you are on campus. I think most of you are at least aware of that. For women, though, it's pretty like broad and general in terms of the guidelines. It's just as long as you're wearing something that is appropriate that you could wear to a job interview, you're good. I don't know. This is, I've noticed every year I teach, I get more relaxed with doing first day stuff like the syllabus. Any questions on the setup or the format of this class?
No.
Okay.
No.
That works. All right. In that case, let's look at some bioinformatics. All right, and so I do have two different PowerPoint slide decks posted. Oh, only that's the wrong course number. I do have two different PowerPoint slide decks posted for this week. We will see how much we have time to get to. This gets weird quick. Let's look at it. All right. Always with the learning objectives, although they never make sense right at the beginning. These are essentially the things that I want to make sure you get out of this lecture. Just beginning, just general things like what is bioinformatics? It will get weirder though. All right. So I get this question a lot. I get the question of what is it that you learn in a bioinformatics class? I get that primarily because we also have the undergrad level bioinformatics. I get the question of is it like really math heavy? Surprisingly, no. Oftentimes not. But what we are going to be looking at is we're going to be looking at a lot of different instances of how you can use a computer to solve some type of biological problem. Questions about genetics, questions about inheritance, things like that. To do that, we need to look at storage of information, retrieval of information, and then manipulation and analysis of information. And that comes in some odd forms. I don't know. I can talk about how weird this class is all day, but I think it'll just make more sense to see it here in a couple slides. If you are like saving and retrieving information on a computer, there are two kind of general ways to go about it. One you're probably familiar with and one you're probably not. If you're saving or retrieving information, you could just use a file. And I think most of you are familiar with that. Like you write a paper for class in Microsoft Word. When you're done, you save it. It gets saved to the hard drive. It gets saved as an individual file. If you want to reload it, you just reload that file. That's not the only kind of common practice, though. The other way that you commonly store information to a computer is to store it to a database. And a database, in the simplest sense, it's not too much different than just what you're working with when you're working with Microsoft Excel. In Microsoft Excel, you're essentially working with just one giant table where each row usually you can think of as some new entry, and then each column is like some category. Databases, kind of the same thing. They're essentially just large tables. Each row is considered a different entry in the database. Each column is just some aspect of that entry. So, like for example, if you wanted to save a gene sequence to a database, you would write to that database. The gene sequence would become a new row in that database. And you might have categories in there like the actual sequence itself. That would be something that would go in one column of that row. And then maybe you would have, say, like the genus and species name of the organism it came from. That would be a different column in that row. Databases, you don't work with so much generally, just on like a casual level, like if you're just working with your home computer. If you're working in a professional capacity, it's the type of thing that you're usually going to have to start interacting with. So a lot of bioinformatics work is all about dealing with databases, and we'll look at that as we go. there's some really common public databases that you can pull information from to kind of help you solve biological problems. When it comes to databases, they come in different flavors. We have things that are referred to as primary databases. Primary databases, the only type of information that's being stored is just what we consider just raw information. It has not been manipulated in any way. It's just however it was recorded. If what you are working with is just something as simple as nucleotide sequences or amino acid sequences, no other information you're really dealing with or concerned about, primary databases type of database you'd be using. In biology, those are cool and all, but they are not all that useful. In biology, most likely you will be working with what is referred to as an annotated database. Annotated databases have the information from a primary database. They have the raw information, but then they have a lot of additional information added. So like if you were dealing with the full genome sequence of an organism, if you just wanted the sequence, just the A's and T's and C's and G's of the DNA sequence, if that was all you wanted, you could deal with a primary database. But generally, that information on its own is not all that useful. What you usually want is the genome sequence, plus knowing where in that genome sequence you could find, say, the start codon and the stop codon of all the individual genes, where you could find the promoter sequences for the individual genes, things like that. And so annotated databases contain the raw information, plus all this extra information that tells you useful things about the raw information. So before things are said and done over the next 8 weeks, we will take a look at some ANA databases, databases where you can get some useful biological information. And then there's a third type. Secondary databases are what we'd call meta databases. Meta databases, usually just a database that's sort of a database of databases. You want to look up, say, a gene sequence in an organism, you could go to a meta database, and that would probably tell you something like where in like 7 or 8 other public databases you could go to get information on that gene sequence. Meta databases are sort of like search engines of databases. They tell you where else to go to find useful stuff. All right. Here is a word I am 99% certain you have never heard before. An accession. One of the things that we will have to look at then is accession numbers. And generally, when you're working with the database, where every row in that database is some new record, individual rows, individual records get some unique identifier. It is a way to make sure you are looking at the correct record. It has to be unique. It has to be something that is not repeated anywhere else in the entire database. It's just you only see it with that row. And so it's a unique identifier that's given to individual database records. People usually refer to it as an accession number, but it almost never is just a number. What we can see here on the left, let me use the pointer. In the parentheses right here, these are individual accession numbers. And if you notice, they actually begin with letters and an underscore. So like NM underscore and then blah, blah, blah. Unique identifier, but it identifies this record here to where if you know the accession number for what it is you're looking for, you can go right to it in the database. All right. If you were familiar with writing computer programs, this first word on here would be something you learned a long time ago. If you're not familiar with writing computer programs, here is your introduction to this term. The term string. A string is any sequence of characters. Strings generally can be text, like terminology right here. This is technically a string. It is a sequential collection of different characters, like T, E, R, M, etc. It can be any series of characters, though. You could have a number thrown in here. So, like if we had terminology 5 for some reason, boom, this whole thing is a string. It could be this. It could be a symbol, one or more symbols in here. Terminology smiley face, this is still a string. But any collection of characters or symbols, that's a string. I bring that up because there are some file types we will be working with in here, and the FASTA file type is the most common file type in biology. And FASTA file types it's a type of file for storing strings, sequences of characters. That makes sense because a lot of things you're looking at in biology essentially are sequences. If you're studying DNA and you're studying genetics, you're dealing with a sequence of nucleotides. If you're studying proteins, you're dealing with a sequence of amino acids. You can save that information. You can always save it to a database or call it back up from a database. But if you want to save it to a file or load it from a file, FASTA file format is the big thing that you're dealing with. I know it looks like FASTA, but it ain't. FASTA, this dates all the way back to the 1980s. There was this guy who came up with a computer program called FAST, and he came up with then a couple of like individual file types for working with this in the 1980s. Like FASTN was for nucleotides. He came up with DNA one, but I forget the letter for DNA specifically. And then he came up with just FASTA. The A is for all. And so the FASTA file format is just a file format for storing any general string. It doesn't really matter the type. So we're going to be looking at these FASTA, FASTALL file types. And we're going to be looking at them right now because here is actually how information is stored in a FASTA file. It is human readable, which means that you could take one of these files and open it up like in Microsoft Word or in Notepad or something, and it would be legible. That's not every file type. If you were to take like an MP3 or a video file or something and open it up in Microsoft Word, it would just be complete gibberish. Most of it would just be symbols. But if you were to open up a FASTA file in like a word processor, you'd actually be able to read it. And it always works like this, where the first line of the FASTA file is going to have this greater than symbol. And then all of the information that comes after that on the first line is really just there for the person looking at this information. The computer kind of usually ignores whatever is on this first line. Whatever is on this first line, there are no rules on it. Whoever made this file can put anything they want on this first line. Even though there are no rules, though, there are some general conventions that people use. And like when you look at this example right here, I can tell you this. Let me change colors. This first thing in here, the GI, the first bit of info that comes after the greater than symbol, this is telling you something about what database this came from. This is information essentially you can also find in this GI database, whatever database that is. The next thing on here, accession number. So it's telling you in that database, what is the accession number that you could use to look for this. And then after that, we get this. Or you could also find it in this other database. Here's another database that contains this record. Here is the accession number in that database. And then finally, the last bit of information it gives you is, what is this? And it's telling you this sequence comes from Homo sapiens, so it comes from humans. It is the hemoglobin beta subunit. This is the messenger RNA sequence. And then after that first line, everything else in here is a string. And so it's a collection of characters. So if you look, this is all A's and C's and T's and G's. Does that make sense for messenger RNA?
Yes. No? Yes?
Yes and no. What's up with the sequence? Is this an RNA
Oh,
sequence
it
that you're
shouldn't.
looking at?
No, it shouldn't have T's.
Yeah, if this was RNA, it shouldn't have T's. Messenger RNA is the source of this, but from like a laboratory or lab work standpoint, if this is the sequence you're dealing with, probably this is what's called a cDNA sequence. Here, let me type this. See. All right, this is basic biology review. Do any of you remember what cDNA is or what cDNA stands for?
The copy DNA.
Kind of. Actually, I feel like that makes more sense than like the official way of looking at it.
Complementary DNA?
Yeah, that's what cDNA stands for, and I feel like that's like a bad name for it. So yeah, the name for this is complementary. All right. All right, from a laboratory standpoint, how do you get complementary DNA? Just crickets. Nobody.
I want to say from copy the original strand of DNA.
Kinda.
Safe to say we have forgotten.
Yeah, yeah. Miss Saint and Miss Horton, the two of you, I know you had this in classes with me too. All right, this is DNA that you get from this. I assume you can hear me typing on my keyboard, so you know I'm typing something. From reverse transcriptase. What? There were no like ahs or anything? All right, so reverse
I said
transcriptase.
by myself, but
Okay, good. Yeah, reverse transcriptase, it's this enzyme, and it's an enzyme that is capable of making DNA from RNA. You can start with a molecule of RNA, you add reverse transcriptase to it, and it constructs a corresponding molecule of DNA. Generally, if you're dealing with a sequence of something, a sequence of a nucleic acid, and you've gained this sequence information from doing general sequencing technology like Sanger sequencing or something like that. Messenger RNA is like impossible to work with. RNA in general as a molecule degrades right away. It is just this really just annoying thing to work with. DNA though is extremely stable and it's easy to work with. And so if you're trying to get the sequence of a messenger RNA, almost always the first thing that you do is you expose it to reverse transcriptase, and then you sequence the resulting DNA that you get from reverse transcriptase. cDNA is just this general term for DNA that you get from reverse transcriptase. I hate complementary DNA as a name. It doesn't explain anything. But okay, so I don't know. That was just some basic biology review. To refresh your memory, or if you did not know that, to give you some background. All right, so we have our FASTA file format. First line is always just general information. Everything after that's a string. All right. We will be working with some FASTA file format stuff in this class as we go. You will see as we do it. Blah, blah, blah. Oh, yeah, this is in case I didn't say it. It's in the slides. Greater than symbol that tells you, hey, first line, that is just information for the person. Everything else, information for the computer. Here is this, and this is just a handy reference. I am not going to ask you like exam questions so much from this, because who in their right mind memorizes this? But if you're looking at nucleic acid string information, it's really more than A and C and G and T and U. Usually when people are recording information as a string in a FASTA file format, they use these other letters too, just to signify some different things. So like if you have done DNA sequencing and there are maybe a couple of nucleotides in the sequence that the sequencing machine was not able to identify, the machine will just put an N in that position. And so N pretty much just means it's some nucleic acid there, but we couldn't identify what it is. And so it's kind of common to see sequences like A, T, T, C, N, G, something like that, which essentially means right here we had no idea what nucleotide that was. These others, yeah, they get used too. Like an S means we were able to narrow it down to a C or a G, but that was as far as we got it. K, yeah, it's a G, a D, or a U, but we know it's not an A or a C. So these different letters kind of tell you different degrees of how certain we are for what a different nucleotide is. If I ask you anything off of this at all ever on a test, it will be N, just because we're going to see that one a lot. And I just want to make sure you know every time we see an N, that just means essentially a question mark. There's a nucleotide, but we don't know what it is. Oh yeah, and just to drive the point home here. Like this is common type of thing you would see from a sequencing reaction. Oftentimes with DNA sequencing, like Sanger sequencing, you get a bunch of N's right at the beginning. The beginning of a piece of nucleic acid is hard for Sanger sequencing to identify. And so it'd be common to get a sequence like this. All right. We, through this course, and especially as we do stuff as a class in person on Saturdays, we're going to be working with some individual databases. I have to tell you that NCBI is the king of bioinformatics databases. The National Center for Biotechnology Information, that is the government bioinformatics institution in the United States. So NCBI is maintained by the US government. All of their databases are free, publicly available. Anyone with some computer know-how can pull records out of their database and look at like gene sequences for different organisms. I was doing it this morning, and I'm not even lying. I always have like my own bioinformatics side like research projects going. I was pulling records over from the NCBI before class this morning. So Also, I'm a massive nerd, and that's why. But we will definitely be working with the NCBI in this class. For Europe, it is the EMBL, the European Molecular Biology Laboratory, sort of like the European equivalent of the NCBI. They have some good software, and we'll actually be using a little bit some of the software from EMBL. These others ain't bad. So all major things that we'll at least like come back to as this course goes on. These two are kind of notable. The two on the list, Uniprot and PIR. These are the only databases on the list that are just completely devoted to protein sequence. They don't really touch on DNA sequence at all. If we have time, we'll actually take a look at least one protein database, because these are usually pretty good databases for doing 3D modeling, and that always just looks cool and sciencey. Usually in these databases, they store more than just the amino acid sequence. They'll try to store some like 3D modeled shape information, so you can kind of see what the protein look like three-dimensionally. These just look cool, honestly. All right. I swear we're almost to where it gets like really bizarre. Just a couple of more basic things that I want to touch on. One, if you've taken undergrad classes with me, you should have seen this before. If not, this is just good but annoying background information to have. When you're looking at double-stranded DNA and you're looking at the central dogma, so remember the central dogma is this idea that you go from a gene in DNA to then the cell producing a messenger RNA copy of that gene, a single-stranded messenger RNA copy of that gene. And then that single-stranded messenger RNA copy of the gene gets fed into a ribosome, and the ribosome uses the information in the messenger RNA to produce a protein. And that's central dogma, central tenet of biology. That's the way genes work. DNA to RNA, RNA to protein. But when you're looking at the gene, you will always have one strand here that the RNA is built off of when you're making the messenger RNA copy of the gene. And so you'll separate the two DNA strands. You'll have an RNA polymerase come in and start building the RNA, and it will build the RNA by complementary base pairing a nucleotide in the RNA with the nucleotide on this bottom strand here of the DNA. That's how it knows how to build the RNA. So that's what we're looking at in terms of this image. This is the especially annoying part. These two strands of DNA go by different names. So the strand down here that the RNA polymerase enzyme is using to build the messenger RNA and match up the RNA nucleotides, this bottom strand goes by these three different names. Why three? It's because it's science and people have to just keep naming things. Antisense, negative, and template, all for the bottom strand here. For the top strand that's just hanging out, just dangling there, sense strand, positive strand, coding strand. If you notice, though, there's something kind of cool going on here. The bottom strand here, this template strand, this is what the nucleotides are being base paired with, at least right at first to build the RNA. But when you look at the coding strand, it has essentially the same sequence as the RNA. So like there's A in this position, there's A in that position. There's a T here and there's a U there, but U is sort of like the cheap knockoff brand of T. And so it kind of matches up. the coding strand is called the coding strand because it will have the same code, the same sequence as the messenger RNA that is produced. All right, so there's all of this. This is technically background information that you should kind of have seen before. All right. Why do I bring that up? Just because when you're dealing with sequence information, like if you're looking at a FASTA file and you're looking at nucleic acid, so you're looking at a sequence of nucleotides. By convention, the sequence that you're looking at is assumed to be from the coding strand. So if this were a gene, we know right here which of the two strands this is. This is the coding strand. It's that one. It's the one hanging out at the top there. All right. There's one more thing going on with the FASTA files and sequence information in general. Oh, and then Kaiser Live always screws everything up. So if you're looking at sequence information, by convention, it is always being given to you in the 5' to 3' direction. So hopefully that rings a bell. If you remember a molecule of DNA, just one. Yeah, okay. It does ring a bell. Each individual strand, it has a direction. One side of it is called the 5', the other is the 3'. So it's always given to you in the 5 to 3 direction. That seems simple enough, but we'll look at some cases coming up later where this can start to get confusing. Just always remember, if you're reading a sequence, it's 5 to 3. There's almost never an exception to that. All right, so it's the coding strand, 5 to 3. That's enough about storage and retrieval and file formats and things like that. Let's get into the weird stuff. Let's now start talking about manipulating information and analyzing information. Let's do this. Chapter 3. What? Learning objectives. All right. Needleman Wunsch. Here we go. This is where it's going to get weird because we need to start talking about algorithms. But I've been talking for 45 minutes. Let's take our first 15-minute break. Let me see. It is, can I do white? That would look kind of cool. Let's say break until 9:45 to go ahead and take a break until 10 a.m. And when we come back, we'll talk about algorithms. Okie dokie. And I'm back. All right. So let's take a look at some information manipulation and analysis in a second. Because before we start talking about the first algorithm that we will see in this class, yeah, I just kind of want to make sure everyone's on the same page in terms of thinking about DNA mutation. So some basic terms. One of these, single nucleotide polymorphism, which is, I have to admit, like an overly complicated way of saying this. I'm typing and the color's still white. Hold on. There we go. Did that fix it? Yeah. Just overly complicated way of saying a substitution mutation, where you've taken a DNA sequence and you've changed one letter. So single nucleotide polymorphism, or SNP, SNP, same thing as just substitution. You've substituted one letter for another. All right. Let's think about this. Let's say that you take a DNA sequence and you change one letter. You change maybe an A to a C. On its own, that may not have done anything. Only like 1.9% of your DNA is actually genes. The other like 98.1% of your DNA largely is just kind of there. Actually, for the most part, it's transposons. That's a completely different topic. But very little of your DNA is actual genes. So let's say that we changed a letter and we changed a letter in an actual gene. Let's say we had a letter change that was actually in a codon, and now I just want to check this. What do we mean by a codon? What is a codon?
Three little nucleotides.
Yes, exactly. Three letter sequence of nucleotides. Absolutely. Codons are something that really technically only occur in genes. Okay, so what's the big deal about a three-letter sequence of nucleotides in a gene? Why does that have like its own name?
Those are the ones that we need to make proteins.
Yes, absolutely. So an individual codon essentially is instructions for what to put in a protein when a ribosome builds a protein. And a codon itself is just three letters that tells the ribosome which amino acid to put in next. So in the most simple sense, you can think of a gene as just like a series of codons, these groups of three letters, three nucleotides in a row that say which protein to put in. All right, so let's say we changed a letter. We changed a letter within a gene, and specifically we changed a letter within a codon. All right. Now then, by doing that change or that mutation, the change could be what we say is synonymous, or the change could be what we say is non-synonymous. If it's synonymous, or the change could be what we say is non-synonymous. If it's synonymous, we changed a letter, did not change the resulting protein. That's actually possible. There's more than one codon usually that corresponds with each amino acid. So we could have changed the letter sequence of a codon, but it could still actually tell the ribosome to put in the exact same amino acid, and we ended up not changing anything. That would be synonymous. Or we changed it, and now we've changed it to a new amino acid. Now we've changed the protein. So synonymous does not really change the protein. Non-synonymous does. Here are some examples. So let's say we changed the codon AAA to the codon AAG. We had that single letter change. Doesn't matter. Both of those tell the ribosome put in lysine. Another example would be, let's say we changed AAA to AAC with our one letter change. Now it kind of screwed stuff up, potentially. Because we went from a codon that tells the ribosome to put in a lysine to a codon that tells the ribosome to put in an asparagine. We changed the protein sequence. We also have these two options, and this is just kind of to see if you remember it. Let's say that we do have a non-synonymous mutation. We're changing what the codon codes for. We have the option of it could be a missense or it could be a nonsense. Missense is we've changed one amino acid to another. Nonsense is we've changed codon for an amino acid into a stop codon. Nonsense mutations tend to be kind of big deals because we are now telling the ribosome to stop building the resulting protein. And this could be at a location that is maybe extremely far away from the original stop point. Maybe we're just building a much, much shorter amino acid now. Sorry. So we have, for non-synonymous, we have missense and we have nonsense. These terms are doofy, I know, missense and nonsense. If you remember from a genetics class, it has to do with whether or not you have a transfer RNA coming in, and we'll ignore it for now. The terminology is doofy. We also can think of things like this now, and this is going to come into play throughout this course. We have one-letter changes that we can categorize as transitions, and we have these changes that we can categorize as transversions. If it's a transition, we're keeping the chemical type of nucleic acid the same. If you remember, we have purines and we have pyrimidines. All right, quick review. Which nucleic acids are purines?
Adenine and
How about-
guanine, I think.
All right, I heard adenine, yep. And guanine. All right. I think I was able to interpret that from multiple voices all at once in my headphones. Yes. All right, the other one is the pyrimidines. All right, for pyrimidines, it's kind of everything else. C, T, U. All right, so those are the three pyrimidines. If we changed an A to a G, that's just a transition. It stayed a purine. If we changed like a C to a U or a T to a C or something like that, just a transition. It stayed a pyrimidine. If you're converting one chemical group into the other, if you're going, say, from an A to a C as your mutation, you're kind of jumping categories here. You're going from a purine all the way to a pyrimidine. That's harder to do by mutation. And because of that, transitions happen far more commonly than transversions. That is something that we will have to take into account later on. And so it's just kind of something good to keep in mind right now. That some of these mutations, all of these mutations are possible, I'll say that, but some of these happen more frequently than others. And it has to do with whether you're keeping the chemical group the same or not. Do not forget about these either. Insertions and deletions. If you are, luckily these names make sense. Goodbye, Miss Saint. Oh, she is gone. Luckily, if you're just inserting some nucleic acids, that's an insertion. If you're deleting some, it's a deletion. It's pretty straightforward. Final one, and then I swear we're done with mutations. And there's a reason I'm doing this. Remember, if you're inserting or deleting, and you're doing it inside of a region of codons, the number that you insert or delete matters. Because if you say insert 3 nucleic acids, okay, you've made essentially one change, really. If you insert 1 or 2 nucleic acids, you've potentially changed everything, everything from that point down. If you just insert 1 nucleic acid, you throw off the location of the codons all the way down, and you essentially end up changing the codons all the way down. So the number of nucleic acids that you insert or delete kind of matters. If it's a multiple of 3, it's not as big of a deal. If it's not, it's kind of a big deal. Hello, Miss Toussaint. You're back.
Yes, it
All
just
right.
picked me out for some reason. It's just weird.
Okay. Yeah, I was told that you were experiencing technical problems like the last class, so that's okay. All right. Let's finally look at it. What we have here is a mutation. We have a piece of nucleic acid that we started with, that's the before. We have a piece of nucleic acid that we ended with, that's the after. It's a short piece of nucleic acid, but still. Here's the question. What was the mutation?
It deleted GT.
Okay, yeah, we're definitely missing a G, and we're definitely missing a T. All right. Let's think about this. Because it gets a little bit weirder when you start thinking about it. Because looking at this, yeah?
I was gonna say, it's not a substitution mutation because it actually took 2, so it's not one letter. So will that be a frame shift?
Oh, yeah. Oh, I see what you mean. Yeah, this is too shorter. Yeah, if this was part of a coding sequence where you had codons, yeah. There would be a frame shift going on too. So there is the possibility of that. That is something to always kind of keep in mind when you're looking at gene sequences. Maybe. Probably without even thinking about it, there's something that your brain is doing that you don't realize. Your brain's probably doing this. It's probably saying this A in the before is that A in the after. Like that didn't change. And your brain's probably saying, okay, this C is that C. Your brain probably is not doing this, but there's no reason why it couldn't. To say like, oh, that G went through a substitution mutation to become that T. Because potentially there could have been a substitution mutation, and then maybe we just lost these 2 T's, and then maybe this A is that A. I mean, that's a thing. Your brain probably is doing like the simpler case of this, where it's saying, all right, this is this, this is this. But then we have this problem. So like this A is probably that A. Which T became the T in the after? Was it the first T or was it the second T that
But
became the T in the after?
doesn't really matter like what T it is, because either way, I don't have like 2 of them. So
Yeah,
we're gonna change.
in some sense, yeah, in some sense, no. To a large degree, yeah, you went from 2 to 1. Maybe it doesn't matter which T we lost. So we'll say just a T to a T. There is this that your brain is doing. Your brain is kind of making assumptions about homology. And so, do you remember like the word homology back from genetics? It means related through ancestry. Usually when we're looking at homology, we're looking at evolution and things like that, but you can use that same idea for instances like this, where we say this A in the after is homologous to this A in the before. They're related through ancestry, but now it's just ancestry through time, the before and then the after. So we're saying like this A is homologous to this A, the C is homologous to that C. The G is just gone. The A is homologous to that A. The T's, who knows, but maybe doesn't matter. All right. What your brain is doing is your brain is doing an alignment. And an alignment is this, where you take one sequence, maybe a before and an after, who knows, but you take one sequence and you pair it up with another sequence and you make statements about homology, where the way this is aligned, what this is saying by this alignment is this A is that A, these two A's are homologous. We're saying this C is homologous. In this case, this is a deletion. So we're saying the C was deleted. We're saying the G was deleted. We're saying this T is homologous to this C, which means there was a substitution mutation. We're saying this T is homologous to this T, and that A is homologous to that A. By doing an alignment, you're making statements about homology, and you're making statements about what has changed. And if you notice the way this was aligned, it's different than just sort of probably what seemed most obvious on the previous slide, where the way we pair it up matters because the way we pair it up tells us what happened, what the differences are. In this case, it was two deletions and a substitution, going by this alignment.
And how do you know that? How do you get to that conclusion? And if it's algorithms, I'm gonna die.
Brilliant question. And actually, that's what the rest of this slide deck is all about. How do you find the best alignment? How can you make the best guess in terms of what has happened? Yeah. So I will try to answer that question. Oh, interesting. Originally there were boxes around all these, but hopefully you can kind of see what's going on. So let me essentially what I've given you is a whole bunch of individual alignments. And if you look at these, all these are just different possible ways that you could align those two sequences that we saw before, the before and the after. All right. So for all these different possible alignments, you can think of it like this. So I already introduced like, okay, this is all about homology. What letter is homologous to what letter? And that tells you what has changed. You can also think about it like this. Anytime you make an alignment where you pair up two sequences like this, you're essentially producing a hypothesis. And it's a hypothesis about what has changed. So we can view this as 1, 2, 3, 4, 5, 6, 7, 8, 9. This is 9 different hypotheses for what changed between these two sequences. The question on the slide is, of all these 9 different hypotheses, which hypothesis is the most likely? And how can you tell?
Well, that will have to do something like with the mutation. So like from the second box, like going from a G to an A, that will be more common than going from a G to a C. So is that one more likely? Because it's
Actually,
a transition, a transversion.
Yeah, transitions are a little bit more likely than transversions. So that is something that we can bring into this. If you can actually look at like what letter changed to what letter. So that is a possibility with the substitution mutation. That some of these substitution mutations will be less likely, and that can ultimately help you kind of rule some of these out. I will tell you this though, that you can actually sort of get to which one of these was more likely without actually even taking into account at this level yet, without taking into account transitions and transversions. So you're thinking along the right lines, but you can sort of answer this even more simply. I'll also give you a hint. You can kind of narrow it down to just 2 of these. And I'm gonna do this. I'm gonna label these. We'll say this is
I
number
mean, I will
1.
go for the ACTA in just the deletion because it seems the most straightforward.
Yeah, you're thinking about it really along the right lines for sure. So there are actually 2 of these that are probably equally likely.
4 and
Looking at
7?
this, 4 and 7. Yeah, 4 and 7 I would say are the most likely. And you're right. That's the right way to think about it. Think about number of things that changed. And when you look at 4 and 7, it's only just 2 individual mutations that you had to do to get to the before and the after for both of those. Like for 4, we deleted the G, we deleted the T. Done. And that's the all that changed. For 7, we deleted the G, we deleted the T. That's all that changed. All these others, it's more than just 2 changes. Like for this sequence here, we would have had to have deleted the A, deleted the C, and then done a substitution mutation to convert the G to the A, and then done another substitution mutation to convert the T to the C. So this would take 4 changes total to go from the before to the after. The like 9, that would take a lot of changes because this is deletion, 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 insertion, insertion, insertion. You have to think about it like this. Every time there's a mutation, there's a probability of that mutation happening. To get more than one mutation. All right, every time you do a new mutation, you take, let's say, probability of the first mutation, and then you have to multiply that by the probability of the second mutation. If you need 3 mutations, then it's the probability of the first times the probability of the second times the probability of the third, on and on and on, to get to the total probability of that happening. If you want to think about this in terms of numbers, let me pick a better color. Maybe this is 0.1 probability, maybe this is 0.05 probability, maybe this is 0.03 probability. Every time we add in an additional mutation, an additional probability, what's going to happen to the overall probability? Every time we add in a new mutation, the overall probability gets smaller. Okay, because we're multiplying a fraction by a fraction by a fraction by a fraction. And so just going at it from a probability standpoint, just if you want to do it completely mathematically, the fewer changes that you do, the higher the probability. The more changes that you do, the lower the probability. So just going by that, 4 and 7 equally likely. 4 and 7, it's both of these are just two deletion mutations. All right, so we kind of got to that with it. All right, so we're looking at alignments, we're looking at homology. Each alignment is a hypothesis, and you can tell good hypotheses from bad hypotheses by looking at the total number of things that has to change between the 2 sequences, because more change is less likely. Now we bring this into it. If you had to do a large number of these, where let's say maybe you had to compare like 1,000 before and after sequences, that's the type of thing that would quickly get extraordinarily tedious for a couple of reasons. One is that just doing 1,000 of anything is kind of boring. And then also, if you really wanted to check that you were finding the best hypotheses, the best alignments, you would actually have to, for all 1,000 sequences you were comparing, you'd have to do all the possible alignments. And so you'd have to make sure that you kind of wrote them all out and then counted up the number of changes. That is the type of thing where if you had 1,000 of these before and after, any person would just not do it. It would be dull enough that they would never succeed, but a computer could do it all day. And so welcome to bioinformatics. We can take really tedious tasks like that and just give it to a computer, and the computer can do it for us. To be able to do that, to be able to take a task like that and get a computer to do it, we need to tell the computer the exact series of steps that it will have to do to solve that problem for you. And so essentially what we're giving the computer is an algorithm. An algorithm is just a fancy word for the complete series of steps needed to solve a problem. It can be very general. It doesn't have to be like worded any specific way. If it's a computer program, it needs to be in computer code. If you just want to tell a person a series of steps, you could be kind of very general with it. That's still an algorithm. Like telling a person how to do this, you would say like, align these in all the possible ways to align them, and then pick the alignments with the fewest number of changes. That's the algorithm for solving this. So it's like a protocol for solving a problem. And the key here is to find the least number of differences between the two sequences for the best hypothesis. Remember this too. So 4 and 7 are the two best hypotheses. That doesn't mean 4 and 7 are correct. If you don't know for sure what changed and you just have the before and after sequence, technically it could have been any of these. You're just making your best guess based on probability. And probability says 4 and 7 are the best bet. But they could be wrong. It could have been 6. I mean, who knows? But so it's all about finding the best guess, at least at this stage. All right. So in case you didn't get the point, the slide is all about this one's better than that one because this one is 2 changes, where that one is 3 changes. All right. To go from the before to the after. Indel. Oh yeah, by the way, insertion or deletion, general term. It just means you've added or deleted something. That's kind of important when doing alignments because what I gave you was a before and an after. And so if we're saying this is the before sequence A and this is the after sequence B, okay, what happened was a deletion to go from the before to the after for this position right here, assuming you can see the pointer. But you're not always looking at 2 sequences that are a before and an after. We could just be looking at 2 sequences from 2 closely related species. And if you're just looking at like sequence from species A and sequence from species B, there's no clear before and after. When you look at this position right here, all that you know is there was either an insertion of a C or there was a deletion from this position. Like you either added a C or you deleted a C. You just know that there is that difference. And that's why this term indel exists, because when you see a position like this in alignment, you know there was an insertion or a deletion. And so it makes more sense to just shorten it and say, oh, there was an indel. We inserted or deleted something. All right. So 2 indels here versus 2 indels and a substitution mutation here. So here's the thing. If you want to set this up in a computer, there's kind of an easy way to do it. At least to tell which alignment is best from a series of alignments. If you start with a series of alignments that you're already giving the computer, easy way to set this up is just to create a scoring system. And we'll say, we'll check the alignments, we'll go down each position, and we're just going to keep a scoring system going. And every time we have a match where it's the same letter at that position in both sequences, we'll say, okay, the 2 letters match, and we'll increase the score. If there is a mismatch, okay, there was some type of substitution mutation, we'll decrease the score. If there's a gap in the alignment, if there was an indel in the alignment, then we'll decrease the score. So if everything stays the same, we increase. If there was some mutation, we decrease. And then what we would do would be we'd just try to kind of find the alignments that produce the highest. What we would do would be we'd just try to kind of find the alignments that produce the highest scores, the alignments where the most things stayed the same. We're going to begin, I'm going to do a couple of examples, and we're going to begin with like a beginner scoring scheme. And this seems weird, but roll with it. We'll say if there's a match, we need to increase the score. If at a position there's a mismatch, so there was a substitution mutation, it says decrease the score, but really we're going to do that by just like not doing anything to the score. We're not going to increase the score for that position. So we will keep the score just what it was if there's a mismatch. And then if there's an indel and a gap in the sequence, then we will decrease the score by 1. Already, we're kind of making a little bit of an assumption here that is present in our scoring scheme. The assumption is also, once again, about likelihood. We're making the assumption that having a mismatch, which is the result of a substitution mutation, is more likely than having an indel, having an insertion or a deletion. And that is actually correct from a biological standpoint. It's easier to have a substitution mutation happen than it is to have an insertion or a deletion happen. And so we're kind of weighting the insertions and deletions differently than the substitution mutations. All right, so we're beginning with that. Our scoring scheme is kind of making some assumptions and some baked-in ideas from just sort of what we know about the way mutations work. And let's go ahead and look at 2 alignments. And we will say, okay, if we use this scoring scheme with it kind of weights insertions and deletions as less likely than a mismatch, we can go through these and we can just calculate a score. And so, like, if we start with 0 as our score, and then we look at the first position, okay, it's an A and an A, so that's a match. Here, let me write these down. So our score is now 1. We increased it by 1. We have a mismatch, and so we have to subtract 1, so we're back to 0. We have a C and a G, that's a mismatch, but our mismatch is we don't change anything with the scoring scheme, so we're still at a 0. We have a T and a gap, and so we need to subtract 1, and so if we subtract 1 from 0, we're down to -1. And then we have a match, and so with a match, we increase by 1 with our scoring scheme, we add 1, so we're back up to 0. And then an A, and that's a match, and we add 1 to our score, and now we're up to positive. And so we say +1 is the total score for that entire sequence. When we went each position and applied the scoring scheme, starting always with a score of 0, and we just roll with it and add, subtract, or keep the same all the way across. At the end, what we get is a +1. This is showing the same calculation, just kind of a different way of going about it. Just counting up all the matches, the mismatches, the gaps, applying the scoring scheme. It works the other way with the other side. Let's do purple. We start with 0. We got a match, so we go up by 1. We got a match, so we're at 2 now. We have a gap, we subtract 1, so we're back to 1. We have a match, so we're up to 2. We have a gap, so we're back down to 1. And then we have a match, and so we're back up to 2. Same thing. The total score for this would be 2. Or you can just kind of do it this way by adding up all the gaps, mismatches. Either way, the one on this side has a score of 2. The one on this side has a score of 1. The alignment, oh my God, let me clear all this stuff off the screen. The alignment on the right-hand side is better using the scoring scheme. And that is the type of thing that it's easy for a computer to do. It's a simple algorithm. We'd say just start with a score of 0, look at each position, apply the scoring scheme. In the end, the alignment that you did that to with the highest score is the most likely. And if you look at these, it makes sense. This one, 2 indels, 1 substitution. That's 3 changes. This one, just 2 indels, and that's it. Fewer changes happened over here. And so it makes sense this would be the better alignment. And if you do that exact same scoring scheme on all of these, the 2 that we kind of picked out as the most likely just by looking at it, those 2 are the same ones that would have the highest score. So using the scoring scheme, it works. Those 2 get flagged as the most likely hypotheses for the best way to align these 2 sequences. Okay. So that's kind of the way to begin this and to go about it, at least in terms of like thinking of a systematic way that you can sort of solve this, and a systematic way that you could tell a computer to solve this, just keep track of a score. There's a downside to this so far. The downside is if you're doing it this way, where you're just telling the computer, okay, keep track of a score. I'm going to give you a series of alignments. You just tell me which one is the best one. The downside is you have to beforehand come up with all possible alignments. If you're dealing with short sequences, that's maybe not too much of a pain. There are very few instances where you would be dealing with short sequences. Oftentimes, if you wanted to do something like find the alignment between a gene in one species and a gene in another species, you'd probably be dealing with sequences that were thousands of nucleotides long. And if you had 2 different sequences that were each like 1,000 nucleotides long, that's a lot of possible alignments. What if you forgot one? What if you didn't make all possible alignments? I have good news and I have bad news. The good news is a pair of researchers back in the 1970s actually came up with a way to check all possible alignments. Not only that, but to check all possible alignments at once. That's the good news. The bad news is it's weird. And so this is kind of the big thing that I wanted to get to in this lecture. I wanted to get to this algorithm for not only applying the scoring scheme, but also checking all possible alignments. Okay, here's the story behind this. Excuse me while I just take a big gulp of my drink right in the microphone. Sorry about that. In the 1970s, these 2 people, Saul Needleman and Christian Wunsch, they were working for the US military. They were government funded. They were doing computer and biology research. They came up with this algorithm for doing this, for checking all possible alignments and applying the scoring scheme kind of all simultaneously. They named this approach, this type of algorithm, they named it dynamic programming. That name has stuck. That's still what you call the type of algorithm I'm about to show you. The problem is, and they freely admit this, that name doesn't mean anything. Dynamic programming, they picked it because it sounded cool, and they knew from being funded by the military that the military in the 1970s tended to only fund projects that sounded cool. That's it. That's the whole meaning of the name, is it's dynamic, and programming was brand new, and so it'd be like naming something like AI blockchain or something like that now, because it's like it's all new. But all right, so the name stuck. You still call this stuff today dynamic programming. Specifically, what I'm going to show you coming up is now referred to not just as dynamic programming, but it's the Needleman-Wunsch algorithm. So it's the specific form of this that they came up with. And what we're going to do is the algorithm is going to do a global alignment between two sequences. Global means that it's going to check all of the alignments between the entirety of both sequences. All of sequence 1 and all of sequence 2, all possible ways to align the two things. Within limits. Like this is not an alignment that this would do for you. Let's say A, T, C, G. Dink, 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 A, C. Like this is not an alignment that would ever be produced by this algorithm because it's not a reasonable alignment. It's just too far. Like essentially we're saying that all of this was changed. A was deleted, C was deleted, T was deleted, and then a bunch of spaces were put in or something, and these were added. It wouldn't do something like that, but it would find all of the at least like reasonable alignments where we're at least aligning some of the letters. All right. So, and it will do all of it for both sequences. If we have to take the time to state that it's global, that's going to align all of sequence 1 with all of sequence 2, that kind of implies that there are other algorithms out there that don't do global. And we'll look at some later on. Somewhere maybe you just want to align part of sequence B with sequence A. So we'll see those later. But this algorithm is going to do all of both sequences. Also, this Needleman-Wunsch dynamic programming algorithm, it's optimal, which means it will always find the best alignment based on the scoring scheme. Okay. I need to point that out because we're going to look at some things later that are not optimal, but still work. All right, so we have all this as our setup. I'm gonna show you this algorithm. We're gonna use the same scoring scheme that we used before, where it's +1 for a match. We do nothing for a mismatch. It's a -1 for a gap. I'm gonna do these two sequences as our example. They're still relatively short. They're longer than what we looked at before, but still short enough. We kind of know there are going to be some gaps in here because these two sequences are not the same length. And actually, we'll say these two came from two different parts of the DNA of the influenza virus. That's of viruses, they tend to mutate really rapidly, so their DNA sequence changes pretty quickly through time. Let's go through all the steps of this Needleman-Wunsch dynamic programming algorithm. All right, to get all possible alignments. But yeah, I know it's kind of annoying. But I got to introduce like a couple terms right at the beginning. These are terms just from math in general. One term is an array. An array, if you remember, like here's array A. Array is essentially just like a collection of numbers or symbols. A string technically is an array. So it's just a series of characters. Like that's an array. It's just a series of characters, series of numbers or letters or something. So what we've thought of so far is a one-dimensional string. All strings we've looked at have been one-dimensional. It's just a sequence of things. Arrays in general are the same thing. They're just a sequence of things. All of these things, technically, these are arrays. They're all technically strings. They're just collections of symbols. And no, this is not Kaiser Live screwing up the formatting. That's really how I made this. So this still counts as an array. This still counts as a string. This is Kaiser Live screwing up the formatting. But essentially what's going on here is, here, I can help you out. Get rid of that. Get rid of that. This is really all you need to look at. This is a matrix. A matrix is just an array or a string with some dimensionality. And by that I mean, in this case, this is a two-dimensional matrix. It's still a collection of symbols or numbers or characters or whatever. But in this case, they're arranged in rows and columns. Essentially, if you're dealing with a two-dimensional array, it's a string given to you as a table. You can think of it that way. It's just a table where you have different row column positions for the different characters in here. All right. Why am I doing all this? Because to do Needleman-Wunsch dynamic programming, where we find all possible alignments and check to see which one gives you the best scoring scheme all at once, requires you to make a matrix. And so we're going to make a matrix as soon as we come back from break, because it's about quarter till the top of the hour again. Let's take our second break. We're only going to 11:30 anyway, which I think is just enough time. We'll say break until it's 10:47. We'll say 11:02. All right. I will see you when we come back from break. And it's 11:02. Okay, I am back. All right, let's look at this. So we need to finally do this Needleman-Wunsch algorithm. It's this general strategy called dynamic programming. The whole point of it is it's a weird way to simultaneously check all possible reasonable alignments between two sequences and find the one specific alignment that gives the best score. The way we're going to do this, we're going to need a matrix. And to begin it, you set it up like this. So this is essentially an empty matrix, which is just a table. So empty table, just some open cells in it. And we're going to begin by filling in the two sequences that we have, one along the first column here and one along the first row. When you set this up, you always set it up to where these first four cells or first four locations in the table are just left empty. So when we look at this, the sequence along the side, this has 4 letters in it, which means total we need 6 spaces down this column here. So essentially 6 rows so that we have room to put this sequence. This sequence right here has 1, 2, 3, 4, 5, 6. It has 7 letters in it. We need 8, 9. We need 9 columns. Also, we can have this empty 4x4 area. All right, so we're setting up our two sequences, and I'm going to go ahead and tell you the way to think about this conceptually is we have a starting position, which is going to be here, and what the goal is to get to here. When you think about it, this is, let me go back to the pointer. This is sort of the beginning location of both sequences. We're beginning with the first letter. Here's the first letter of that sequence. The goal is to get to down here where it's, this is sort of matches up with the last letter of that sequence and the last letter of this sequence. So we want to find a way to get from beginning of both to the end of both. That will make it global where we kind of cover all of both sequences. Now then, what really we need to do is we need to find the best path, the best way to go from the starting location to the ending location. And when we do this, anytime we move in this direction, it's kind of the equivalent of aligning the letters along here, along the top sequence, with gaps. If we're just moving just straight to the right-hand side, we're moving through this top sequence, but we're not moving through the sequence along the side. So if we're moving through the top sequence, but not moving through the other sequence, it's kind of like we're aligning this with gaps. All right. Other way, gaps. All right. Other way around, if we're moving straight down as our direction, all right, then we're moving through the sequence along the side, but we're not moving through the sequence along the top anytime we're going down. So if we were to start at the starting position, just go straight down, it would be like aligning the letters in the sequence on the side with gaps. Okay, that's kind of the way to think about that if we're moving straight down. So right to the right, we're moving through the top sequence, but not through the side sequence. Moving straight down, we're moving through the side sequence, but not through the sequence along the top. All right. If we're moving, what's a good color? Here we go. If we're moving diagonally, then we're actually moving through both sequences. If we were to move diagonally through here, that's kind of the equivalent of if we align this C with that C, and then we align this A with this G. So if we're moving diagonally, we're actually aligning letters from both sequences then. All right, so we kind of have this idea what our directions mean, and those are really only kind of the only three directions we can go. We can go right, we can go down, or we can just go diagonal. We know there are going to have to be some gaps somewhere in here because these two sequences aren't the right length. And that shows by the fact that we could not just go diagonal and get to that final square. If we just went diagonal, we'd align the C and the C, the A and the G, the C and the C, the G and the A, and then we'd have three letters left over. If we just went diagonal, we would then eventually have to go off to the right to get to that final square. And that would be where we just kind of move through the top sequence, but we didn't move through the side sequence. So it's all about, we're going to have to find the best path through here. The path that gets us from that first square to the final square. And it's going to have to be a path that produces a score. So there has to be some scoring scheme associated with moving through here. And it will have to be the path that produces the highest score. So that's the goal already for this. We have to find how we want to move through the space, and we want to move through the space in a way that maximizes the score that we get. So it is kind of set up like a game in that regard, with a goal to get from one cell to the other. All right, but I still haven't kind of shown you how to do this. Let's look at how to do it. Once you've set up your matrix and you've set it up to where you've put the sequences in along the top and the side, and you've left these 4 empty cells, you still need to do this. You have to initialize some numbers in here. And so if you remember, we need to kind of go from this cell to that cell. That still leaves this row and this column right here that we can do stuff with. And so what we're going to do is we're going to put a 0 here to set this up. It's like setting up a board game. And then as we go off to the right, we're just going to put, we're going to just put the number that came before it subtracted, with 1 subtracted from it. We'll say that. So we start with 0, then we go down by 1, we're -1. We go down by 1 more, we're -2. Same thing along this column right here. This first cell is a 0, but then we just go 1 less than that. So -1 there, 1 less than that, -2 there. We put in those numbers and that sets it up. That gives us some initial set of numbers we can kind of work with. And what we're doing, really, when you think about it, is remember our scoring scheme. Our scoring scheme is if we're matching 2 letters, we increase the score by 1. If we're aligning 2 letters, but there's been a substitution mutation, it's not the same letter, then we keep the score 0. If we're putting a gap in somewhere, then we subtract 1. And by initializing this first row and this first column, that's kind of what we're doing. We're starting with the score of 0, and we're saying, this is what happens if you only move to the right. If you only move to the right, you're only moving through one sequence, you're not moving through the other, so you're including gaps. And so this is like if you were to include gap, 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 gap. Same thing with going down. Going down, you're moving through the sequence on the side, but not the sequence along the top. And so you're essentially aligning gaps. And so this is how you would adjust the score if it was just a gap after a gap after a gap after a gap. But once you've done that, once you've put in your sequences and you've initialized the first row and the first column, now we're ready to get going with it. And we can actually finally start doing this process of starting with this first cell and finding the best path to this last cell. All right. Oh yeah, you can think of it like a map. All right. So this is what we're going to do. We're going to start with this cell here. And eventually we're going to have to check every single cell in here. But as we go through these, every cell that we're looking at, we're going to do this. The rule of the game is we're going to calculate 3 different scores, and we're going to take the best one. So the 3 different scores we're going to calculate are going to be the scores that would result from our 3 different movement options. So remember, we could move to the right, we could move down, or we could move diagonal. We need to figure out which one of those gives us the best score, and that's the number we're going to record in that cell. So let's do this. We're going to do, let's find the first score. The first score we're going to say is our match-mismatch score. What we're going to do is we're going to look at what 2 letters are being aligned at that position. If it's the same letter, we're going to increase our score by 1. If they're different letters, the rule for mismatch is you just leave it the same. You don't do anything to your score. And the score that we're going to be looking at is going to be the one that's up and diagonal. And so let's look at this. If we think about this, for this position, our match-mismatch score is the score if we were to move diagonally through here, which would mean the same thing as aligning the C with the C. And if we have a C aligned with a C, the rule for our scoring system is this is a match. We increase the score by +1. And what we're adjusting is the starting number here, this 0. And so, okay, so for moving diagonal, it would be a C to a C. It would be 1 plus 0. All right. So our first score is 1. All right. We still need to calculate 2 other numbers for that cell just to make sure, just to see what the highest number is. But the number for moving diagonal is 1. The next number we need to calculate is our gap score, but it's the gap score essentially for what would happen if we moved to the right. If we move to the right, we're taking this letter, but we're aligning this letter with a gap because we're moving through the top sequence, but not through the side sequence. So by moving that direction, it's a gap. Our gap score is we subtract 1. And in this case, what we're subtracting 1 from is the number over here, the number to the left. So if you move diagonal, the score that you're concerned about is the score that's diagonal from you. If you move to the right, if you move horizontally, the number that you're concerned about is the number that's horizontal from you. So we'll say this is the score we're starting from. We subtract 1 from it. Our second number that we've calculated is a -2. Okay. We have one more number that we need to find, and then we'll find the biggest number of the 3. This is our second gap score. This is the score that we would get from moving down. If we're moving down, we're moving through this sequence, but we're not moving through the top sequence, so it's the same as aligning the C with a gap. Since we're putting a gap in for this movement, we subtract 1, and since we're moving vertically, the score that we're adjusting is the score that is vertical from us. So the number that we're starting with is a -1. We subtract 1 from it, we get a -2. We have now calculated our 3 numbers. And when you think about it, our 3 numbers were, we had a 1, that was when we moved diagonally. When we moved diagonally. We had a -2, that was when we moved horizontally. We had a -2, that was when we moved vertically. So that's the rule that we apply to every cell. We calculate those 3 numbers, and then we pick the highest one. And in this case, and this is just to make sure that people are out there, and I'm not talking to myself at this point, what's the highest number of the 3? Oh God, I'm just talking to myself.
One? I'm so confused.
Yes. I also wanted to check that. Miss IU, did you say something? I saw your microphone light up, but I didn't hear anything. Or is your microphone busted again? Oh, no, your mic's not working. Okay, so that explained that.
See, Dr.
Yeah.
Copes, it was bad. Like yesterday
Yeah.
was weird. I had to refresh.
Oh, yeah, I saw you. You screamed one. Yeah. So, yeah, I saw the microphone light way up. Yeah, Miss Tassan, I've seen you like log out and log back in a couple of times. That's okay. I'm monitoring like what
Like,
everybody's doing in terms of.
I have my
Yeah.
laptop and I have my mobile phone, and it's like I'm playing back and forth to see what's going on because one drops, the other one don't drops, and it's like, what is
Yeah.
everyone? Is it something in the system?
All right. Let's just make sure everyone's on the same page here. So we had to calculate 3 different scores. We're starting here, this first cell, and we had to find 3 different things, 3 different numbers. The first number was kind of associated with a diagonal movement through our map here. If we're moving diagonal, that's the same thing as aligning a letter with a letter, and we apply the scoring scheme that we use if we're matching up a letter with a letter. In this case, the 2 letters it's a C and a C, and so that's a match. And so according to our scoring scheme, if we're aligning 2 letters and it's the same letter, that's a match, we increase the score by 1. And the score that we're concerned about is the score that's diagonal from us. We're concerned about this. So 0 plus 1, that gives us a 1 as our first number. Second score is the score if we're moving horizontal. Let me switch colors so it's not completely wonky. If we're moving horizontal, a horizontal movement or a down movement, either one, we're including a gap. So if we're moving horizontally, it's like we're putting a gap in the sequence. It's like we're keeping this C here, but we're trying to align it with a gap. If we're putting in a gap, then it's -1 always. That's the way it goes by your scoring scheme. But it's -1 from the number that is horizontal from us. And so it's going to be this -1. So -1 minus 1 is -2. All right, that's the second number that we calculated. We still need to do a third number. The third number is the equivalent of moving down. If we're moving down, that's a gap. It's like keeping this letter but aligning it with a gap. If we're doing a gap movement, then we subtract 1. That's our scoring scheme. And we subtract 1 from the number that is vertical from us. And so it would be 1 subtracted from this number. -1 minus a 1 is -2. That's the rule. That yellow on the white really looks horrible. That's the rule for this. In every cell, we have to check those 3 things: diagonal movement, horizontal movement, vertical movement. We get a score for each. We take those 3 numbers, and then the number that is the highest, that's the winner, and that's the number that we end up putting in that cell. 1, -2, -2. For those numbers, what's the highest number?
One.
Yes, it's the same thing that Miss IU screamed, and I couldn't hear it. Yes, Miss Thompson, exactly. All right, just making sure everybody's on the same page. All right, so 1 is the highest number. So that's the number we keep. We disregard all the others. And so this is what we do. Let me clear all this garbage. Ultimately, what we'll put in here is we'll put a 1, and we record the direction that we moved. So remember, we checked diagonal movement, horizontal movement, vertical movement. the diagonal movement 1, the diagonal movement was the winner. And so in that cell, we put actually 2 symbols. We put the 1, the highest score, and then we put the direction that we moved through that cell. Okay. So far, so good. Now we just do all of the other cells, which is tedious as hell, and therefore the type of thing that is perfect for computers. There is a rule. There's one more rule to this game, which is like the worst drinking game ever. The one more rule is as you move through these cells, there's a pattern to which cell you go to next. We're always moving through them like this. This is actually referred to for some reason as an anti-diagonal, but essentially it will kind of work like this. We do this first cell. The next cell that we calculate the 3 scores for will be this one here, if you see my pointer. After this one, then we go to that one, and we've completed that anti-diagonal. Once we're done with this, and we've recorded the highest number and the direction, then we go to here, then there, 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 then there. And so we fill them in like that. We fill them in as these anti-diagonals all the way until the last cell that you do this set of rules for is this cell right here, the bottom one. And so we fill them all in like that. I think, let me check. No, I don't have it for you guys. I will get some practice posted. We will definitely be doing this in person on Saturday. Oh yeah, anti-diagonals. So you fill them in this pattern. If you were to do that, if you were to, oh, let's work through a couple of cells together according to the slides. If you were to do that, you'll eventually get like this whole map made out. Let's do one more just to make sure everyone's on the same page. Let's do the next one up going by the anti-diagonal system would be this one here. So that row, that column. Remember, our scoring scheme is it's +1 for a match, it's 0 for a mismatch, it's -1 for a gap. All right. For this cell, if we're doing diagonal movement and we're checking our match-mismatch score, we have an A and a C. Is this a match or is this a mismatch? Yeah, exactly. It's a mismatch. So 0's the rule, the scoring scheme. We're not changing anything. That's why it's in all caps. Okay, thank you. So it's a mismatch. We're going to essentially just keep our previous score. What number do we keep? That's the question. Where do we look? What number are we adding 0 to? Remember, it's diagonal movement is what we're checking first. Yeah, the -1. So if it's diagonal movement, the score that we're adjusting is the score that's up and diagonal from the cell. So it'll be -1 plus 0, which is still -1. And so for our first number, that's what we record. That would be a -1. We got to it by diagonal movement. All right, next one is horizontal. And anytime we're moving horizontal or vertically, it's a gap. And so we know what we're going to do for that one. We're just going to add -1. All right, so for this cell right here, if it's horizontal movement, we're adding a -1, but we're adding it to what number?
1.
to the 1, exactly, to the score that's horizontal from it. So 1, and we're adding a -1 to 1. That gives us what number? No. 1 plus
0.
-1. Yes. 0. Yes, thank you, Miss Thompson. So yeah, 0. So we'll record that. It was diagonal movement. Last one. Still a gap. We're just going vertically in this cell. It's going to be, we add a -1, but we add a -1 to what number?
-2.
Yeah, exactly. The number that's vertical from here. So to the -2. So -2 plus -1 is, I like this math, -3.
-3.
Yes, Miss Ayu.
3.
Yes, Miss Horton. Yes, Miss everybody. Yes. So yeah, -3. All right. So those are our 3 scores. -1, 0, -3. Of those 3 numbers, which is the highest?
0.
0. Yes, I feel like this is 2nd grade mathematics. Yes, that is the higher number of those 3. And that's what we would record in that cell. We would record the 0 and the direction of movement that produced that highest score. It's just that same pattern then over and over and over again. Like we would go to this cell, then to that one, then to this one, then to that one. We have like 1.5 minutes remaining, and I still kind of haven't wrapped this up. And so this is what I'm gonna do. If you were to do all of these, this is the pattern that you would get. If you notice there's something weird going on for some of these, like this one here, it's 2 different directions. You do that whenever there's a tie. If your highest number, you could get to that highest number from 2 different directions. You just have to record both directions. So some of these, it was a tie to get to that highest number. There's also something else that you can kind of spot by looking at this, and that is the arrows were drawn kind of going backwards. And the reason for that is to find the final best alignment, what you do is you start at the end and you trace back. And so you can say, all right, it was movement in this direction, then it was movement in this direction, then it split. And so already right here, this tells us we have more than one best alignment. We have more than one thing that gave us like the highest score. So we could have gone this direction, then this direction, then this direction, then that direction. That would get us back to the beginning. Or we could have gone this direction. Oh, and we have another split. Okay, so now it's like 3 different paths through here. We could have gone back, and that would have got us there. Or we could have actually gone all the way up to here. And essentially this takes us, once you hit this, then you just go straight over. So we have 3 different paths through here that ended up being the best. That means it's a 3-way tie between 3 different alignments. Let's see. So this kind of shows it to you right here. This I actually got from a textbook. So this is sort of the figure from the book. When you think about it, and this is how I'm going to end it, and so you see sort of how it's done. We ended up with 3 separate paths back to the beginning. And when you write out the alignment, you actually start backwards. So we say, all right, the first thing we did was we went horizontal. Going horizontal is the same thing as moving through this top sequence, but not moving through the side sequence. So horizontal is the same thing as taking this letter, that T, and aligning it with a gap. And if you look, that's what we have here at the end of the sequence. For all 3 of these best paths, It's a T in the top sequence and then aligned with a gap. You go to the next cell over and it's diagonal. Diagonal is the same thing as aligning this letter and this letter. And that's what we have at the next position over in all these sequences, is the A aligned with the A. You go to the next one over, and now here's where it splits, where we have more than one option. We could either go diagonal, which is the same thing as aligning the T and the C, which is what we have here and here. Both of those align that T with that C. Or we could go horizontal. That's the same thing as aligning the T with a gap. And that's what you have as the third option down there. You have this T aligned with a gap. And if you just keep working your way over and applying that rule of diagonal movement is you're aligning a letter from both sequences. Horizontal movement is you're taking a letter from the top and aligning it with a gap. We don't have any verticals in here, but vertical movement would be taking a letter from the side and aligning it with a gap. But you do that and you trace it back and you trace back all the paths. That gives you all your alignments. And the way this is set up, it is guaranteed that the one alignment or the top group of alignments that you get, the paths that you get out of this, will be the best scoring possible alignments out of all the possible alignments. It's annoying, it's tedious, it's hard to remember all the rules. It's what we'll talk about at the beginning of class on Saturday. With that said, I went 3 minutes over. I apologize. I wanted to get to this part of it. Miss Thompson, if you don't mind, stick around just like if you can, just stick around like 3 minutes after class is over and everyone's signed out. It's just because you're a new student, and I just want to say a couple.
Okay.
Okay. Everybody else, get out. Everybody else, I will see you on Saturday. If you have any questions about any of this material, because it's weird already, send me some emails and stuff like that, and I'll get back to you. Trust me, we'll go back over this and we'll pick back up with it Saturday morning at 9 when class starts. I don't know. Anybody have anything before you take off? Or are you good?
I'm good.
All right. I will see you on Saturday.